Hi, this is Dr. Wilma, Professor and HOD of Biochemistry from Bowring and Lady Curzon Medical College and Research Institute. I welcome you all to this competency acid-base balance. Life is a struggle not against sin, not against money power, but against hydrogen ions. Yes, rightly said by H.L. Mencken, our body is struggling against these hydrogen ions to maintain acid-base homeostasis. The human body can be described as a complex system which consists of several levels. At the chemical level, acids and bases are one of the essential compounds upon which all biochemical processes depend. Biochemical reactions occurring in the body are extremely sensitive to even small changes in the acidity or alkalinity of the environment in which they occur. The acid-base homeostasis is essential for cellular viability, enzymatic reactions, protein conformation, and central nervous system functions. There are several other reactions which are entirely depending on the acid-base status. Just imagine that you're posted in an intensive care unit and you'll be given this report to interpret and manage the case. So what you're going to do? So the appropriate diagnosis and management of acutely ill patients requires prompt and accurate interpretation of the acid-base status. Without a basic and thorough understanding of acid-base chemistry can lead to erroneous interpretation and may even hamper patient management. The biochemical functions are modified greatly by any departure from narrow limits of normal cellular and extracellular acid-base status. For these reasons, the acids and bases that are constantly formed in the body must be kept in balance. So, let us discuss about the acid-base balance now. What are the specific learning objectives of the competency acid-base balance? So at the end of this session, I expect you to know about the definition of acids, bases, pH, pK, buffers and Henderson-Hasselbalch equation, and also to describe the regulation of acid-base balance in the body. Before going to the details of acid-base balance, let us try to understand what are the basic concepts which are required for discussing the acid-base balance. So the basic terminologies are acid, base, pH, buffer, pK and Henderson-Hasselbalch equation. So what is an acid? An acid is nothing but a molecular species which can donate a proton, and base, it's a molecular species which can accept the. According to Bronsted and Thomas Lowry, acid donates a proton and the base accepts the proton. Here we can see the example of carbonic acid donating a proton and bicarbonate accepting the proton to form carbonic acid. Dr. Sorensen expressed pH as the negative logarithm of hydrogen ion concentration. So what do you mean by pH? pH is nothing but the Pondus hydrogeny, means potential hydrogen. So when the hydrogen ions are placed in an aqueous medium, that means water containing solution, it gives rise to acidity to the solution in which it is placed. So pH is nothing but negative logarithm of hydrogen ion concentration. So there is an inverse relationship between pH and the hydrogen ion concentration. As the pH increases, the hydrogen ion level decreases. As the pH decreases, hydrogen ion concentration increases. Here we can see the pH scale ranging from 0 to 14 and 7 is the neutral pH. At pH 7.4, the hydrogen ion concentration is 40 nanomoles per liter. The normal arterial blood pH is 7.4 plus or minus 0.05. The normal venous blood pH is lesser than the arterial blood pH. So the venous blood pH range is 7.38 plus or minus 0.02, which is lesser than the arterial blood pH. So the normal arterial blood pH ranges from 7.35 to 7. So what is pK? pK is nothing but it's the pH at which the weak acid is off dissociated. That means 50% is in the associated form, 50% is in the dissociated form. Consider a weak acid here. 50% of this weak acid HA is in the form of undissociated form and 50% is in the dissociated form. So the pK of the reaction is 6.1. That means it's the pH at 6.1. The weak acid is in the 50% dissociated and 50% in the undissociated form. So what is the use of the pK? This indicates the buffering capacity. So the buffering capacity is optimum when the pH is equal to pK. So if the pK is equal to pH, then the buffering capacity is optimum. Otherwise, if the pH is equal to pK plus or minus 1, then it has got effective buffer capacity. What is buffer? Buffer is a solution which consists of combination of weak acid and its conjugate base. This solution resists or minimizes the change of pH when acid or a base is added to it. The effectiveness of the buffer depends on the pH of the environment in which it is placed, buffer concentration, and also the pK of the buffering system. Already we have discussed about the pK. So the buffering capacity is optimum when pH is equal to pK, and it is an effective buffer if pH is equal to pK plus or minus 1. The terminology buffer was introduced by Walter Nernst for carrying out the biochemical reactions. Here we can say at the arterial blood pH 7.4, the weak acid carbonic acid has got pK of the reaction 6.1. We know the terminologies, the definitions. The pH is nothing but negative logarithm of hydrogen in concentration. Acids donates the proton, bases accepts the proton, strong acid dissociates completely, weak acids incompletely dissociates, and the pK of the reaction is nothing but it's the pH at which the weak acid is off dissociated and alkali reserve. So we'll discuss this later. And the buffer solution, buffers means it's a solution which minimizes or resists a change in pH when acid or bases are added to it. Henderson-Hasselbalch equation is another aspect which is required to understand acid-base balance. Consider a reaction here as HA as weak acid which dissociates to form hydrogen and the base. According to law of mass action, the rate of the reaction is directly proportional to the product of the molar concentration of the products to the product of the molar concentration of the reactants is constant. So when you simplify this and substitute negative logarithm on both sides, you get pH is equal to pK minus log weak acid to base. When you again simplify this by reversing this, it becomes the positive logarithm of base to acid. So pH is equal to pK plus log base to acid. Henderson-Hasselbalch equation indicates the relationship between three parameters pH, pK and ratio of conjugate base to the undissociated acid. It enables one to relate quantitatively the changes in base, acid and the pH. So if you know the concentration of the pK and the ratio of base to acid, you can calculate pH, and if you know the pH value and the pK value, you can calculate the ratio of base to. After knowing about the basic terminologies which are required to understand acid-base balance, let us know what are the major threats to the pH of the body fluids. The major threats to the pH of the body fluids are acids which are formed in the metabolic processes. The metabolic acids can be conveniently divided into volatile acid and the non-volatile acid. Volatile acid is carbon dioxide, which is a major end product of the oxidation of carbohydrates, fats, and amino acids. It can be regarded as an acid by virtue of its ability to react with water to form carbonic acid, which in turn can dissociate to form hydrogen and the bicarbonate. Because it is a gas, carbon dioxide, often termed as volatile acid, and we have the non-volatile acids. Again, under this we have fixed acids and the organic acids. Fixed acids are sulfuric acid and phosphoric acid. Sulfuric acid is the end product of oxidation of sulfur containing amino acids like methionine and cysteine, while phosphoric acid is formed in the metabolism of phospholipids, nucleic acids, phosphoproteins, and phosphoglycerides. In contrast to carbon dioxide, sulfuric acid and phosphoric acid are non-volatile and have therefore been termed fixed acids. Then we have the organic acid, the common organic acids which we always discuss are lactic acid, acetoacetic acid, and beta hydroxybutyric acid. When these acids, which are volatile and non-volatile acids, are poured into a body, what is the defensive mechanism which plays a major role against changes in hydrogen ion concentration? There are three primary systems that regulate the hydrogen ion concentration in the body fluids to prevent acidosis or alkalosis. The first one is buffer systems of the body fluids. Second one is the respiratory system, and the third line of defensive mechanism is by renal system. The buffer systems of the body fluids reacts within a fraction of a second to minimize these changes. Buffer systems do not eliminate hydrogen ions from the body or add them to the body, but only keep them tied up until balance can be re-established. Respiratory system, the second line of defensive mechanism, regulates the removal of carbon dioxide from extracellular fluid within few minutes to 1 or 2 days. But the third line of defensive mechanism, that is the renal system, can eliminate the excess acid or base from the body. Although the kidneys are slow to respond compared with the other defensive mechanisms, they are the most powerful and long-term regulators of the acid-base regulating systems. Here we have the explanation for the balance of hydrogen ions in the human body. We have the production versus buffer systems of the body fluids as well as the other defensive mechanisms, that is by the respiratory system and the renal system. Production of hydrogen ions, approximately 20,000 millimoles per day by the tissue respiration and 80 millimoles per day by the non-volatile acids, are buffered by buffer systems of the body fluids as well as the respiratory system by elimination of carbon dioxide and handling of hydrogen ions and the bicarbonate by the renal system. So we have the defensive mechanisms against changes in hydrogen ion concentration. The first line of defensive mechanism is buffer systems of the body fluids, and the second line of defensive mechanism respiratory system, and the third one, which is the most powerful and long-term regulator of hydrogen concentration, is renal system. Let us discuss one by one. The first line of defensive mechanism, buffer systems of the body fluids. Let us know in detail about buffer systems of the body fluids. There are various buffer systems which play a major role in maintaining the acid-base balance. So in the blood we have bicarbonate buffer system, hemoglobin buffer system, protein buffer system, and the phosphate buffer system. In the CSF and ECF, that is extracellular fluid, we have bicarbonate buffer system, protein buffer system, and phosphate buffer system. In the intracellular fluid, we have the similar ones as in case of ECF, protein buffer system, phosphate buffer system and bicarbonate. But in case of urine, the renal system, we have phosphate buffer system, bicarbonate buffer system and the special buffer system, that is the ammonia buffer system, which is specific to the renal system. So though ammonia is toxic to our body, it acts as a buffering agent in the renal system. Bicarbonate buffer system, which is one of the buffer systems of the body fluids, consists of two components, weak acid, that is carbonic acid, and the base bicarbonate. Carbonic acid is formed in the body by the reaction of carbon dioxide with water in presence of the enzyme carbonic anhydrase, which is especially abundant in the walls of the lung alveoli as well as the epithelial cells of the renal tubules. When you consider the Henderson-Hasselbalch equation, that is pH is equal to pK log base to acid, here the bicarbonate concentration in bicarbonate buffer system is 24 milliequivalents per liter and the carbonic acid is calculated based on the values of partial pressure of carbon dioxide and the solubility coefficient for carbon dioxide, because in the body fluids carbonic acid is in equilibrium with the dissolved carbon dioxide in water. So the concentration of carbonic acid is 40 multiplied by 0.03, which is equivalent to 1.2. So when you substitute carbonic acid value, that is 1.2, and the bicarbonate 24, we get 20 by 1. So the log of 20 is 1.3 and pK of the reaction is 6.1. So the pH of the reaction is 7.4. So this is the normal arterial blood pH, which indicates that at this pH 7.4, the concentration of bicarbonate is 20 times that of the carbonic acid. Though the reaction of this bicarbonate buffer system is having pK of about 6.1, which is not representing the optimal body buffer, but the bicarbonate buffer system is regarded as the most important buffer system in the body because the concentration of its components, the bicarbonate to carbonic acid, is 20:1. Hence, it is considered as an important buffer system in the body fluid. And also, the other aspect is it is handled by two systems, that is renal system and the respiratory system. Bicarbonate is handled by the renal system and the carbonic acid by the respiratory system. So let us see how these two systems are independently regulating the bicarbonate buffer system. When a strong acid is added to the bicarbonate buffer system, the bicarbonate component of the bicarbonate buffer system buffers the hydrogen ion to form carbonic acid in presence of the enzyme carbonic anhydrase, which dissociates to form water and the carbon dioxide. This carbon dioxide stimulates the respiratory center and increases the respiratory rate so that there is expulsion of carbon dioxide from the lungs. At the same time, the renal system maintains the bicarbonate level. When a strong base is added to the bicarbonate buffer system, the carbonic acid, the weak acid component of the bicarbonate buffer system, buffers the base, strong base, and forms the water and bicarbonate. This bicarbonate is eliminated by the renal system. At the same time, the carbonic acid level is maintained by the respiratory system. With this explanation, we can say that though the bicarbonate buffer system does not represent an optimal body buffer, but the bicarbonate buffer system is regarded as the most important buffer system in the body because the concentration of its components, the bicarbonate, which is 20 times that of the carbonic acid, and the components can be independently regulated, that is carbon dioxide concentration by the respiratory system and the bicarbonate by the renal system. So when the disturbance of acid-base balance takes place due to the primary change in the extracellular fluid bicarbonate concentration, then it is referred to as metabolic acid-base disorder. When the primary change in PCO2 occurs, then it is referred as respiratory acid-base disorder. The phosphate buffer system is mainly found in the intercellular fluid with the pK of the reaction 6.8, which is close to the pH of 7.4. Hence, this is potentially a good buffer at blood pH. However, phosphate buffer in plasma is low. Hence, it is not an important buffer in the extracellular fluid. It plays a major role in buffering renal fluid and the intercellular fluid. The main elements of phosphate buffer system are dihydrogen phosphate and the monohydrogen phosphate. The ratio of base to acid phosphate is 4:1. So the components of phosphate buffer system are dihydrogen phosphate and the monohydrogen phosphate with the pK of the reaction 6.8. So whenever a strong base is added to the phosphate buffer system, the acid component, that is the dihydrogen phosphate, buffers the strong base, and when strong acid is added, the monohydrogen phosphate, the component of the phosphate buffer system, buffers the strong acid. Here we have the Henderson-Hasselbalch equation. Let us find out the ratio of base to acid in this buffer system. The pH of the reaction is 7.4, that is arterial blood pH, and the pK of the reaction is 6.8. So when you simplify this, we get 0.6 is equal to log base, that is monohydrogen phosphate to dihydrogen phosphate. So antilog of 0.6 is 4, and we have the ratio of base to acid is 4:1. That means the monohydrogen phosphate, the base component, is 4 times that of the dihydrogen phosphate, that is the weak acid component of the buffer system. The phosphate buffer system is important in buffering intracellular fluids because the concentration of the phosphate in these fluids is many times that in the extracellular fluid. Also, the pH of the intercellular fluid is lower than that of the extracellular fluid and therefore usually closer to the pK of the phosphate buffer system compared with the extracellular fluid. Proteins are the intracellular buffers along with phosphate buffer. Buffering capacity of proteins depend on the pK value of ionizable side chains. The most effective group is histidine imidazole group with a pK value of 6.1. Proteins are among the most plentiful buffers in the body because of their high concentration, especially within the cells. But they have limited capability of buffering in blood because most of the plasma proteins are present only in quite small concentrations. But the albumin is the most abundant plasma protein and does contribute significantly to the blood buffering. An effective protein buffer is hemoglobin, which is an important buffer in RBC, and it has got an important role in respiratory regulation of pH. Hemoglobin has approximately 6 times the buffering capacity of the plasma proteins in blood due to its high concentration and its 38 histidine residues. The pK value of oxyhemoglobin is about 6.6, while that of the deoxyhemoglobin is about 8.2. Thus, at pH 7.4 of normal blood, both are equidistant from their pK values, and so both are equally good buffers. In prolonged acidosis, phosphate buffering can become an important buffering system in the bone. Calcium phosphate, which is present in the bone as hydroxyapatite in the inorganic part of the bone, is relatively insoluble. However, the solubility is greater at lower pH. Hence, if the pH falls below the normal, some calcium phosphate, which is present in the bone, goes into solution and the plasma levels of both calcium and the phosphate increases. The increased phosphate is thus able to buffer hydrogen ions. Here we can see the acceptance of hydrogen ions by the phosphate to form monohydrogen phosphate. So the buffering of again hydrogen ions by the monohydrogen phosphate to form dihydrogen phosphate. This calcium phosphate in bone can be regarded as an alkali reserve which is mobilized in response to reduced pH. So the percentage contribution of buffer systems of the body fluid are as follows: 64% by the bicarbonate buffer system, 29% by the hemoglobin buffer system, 6% by the protein buffer system, and then 1% by the phosphate buffer system. Hydrogen ions are common to the reactions of all the buffer systems of the body fluids. So, whenever there is a change in hydrogen ion concentration in the extracellular fluid, the balance of all the buffer system changes at the same time. Here we can see the bicarbonate buffer system, phosphate buffer system, hemoglobin buffer system, and the other protein buffer systems. This phenomenon is called as the isohydric principle. The second line of defensive mechanism against acid-base disturbances is control of extracellular fluid carbon dioxide concentration by the lungs by increasing or decreasing the rate of respiration. The rate of respiration is controlled by the central chemoreceptors and the peripheral chemoreceptors. Central chemoreceptors are located in the respiratory center, that is the medulla oblongata, and the peripheral chemoreceptors are located in the carotid bodies and arch of aorta, which are very sensitive to changes in the pH and pCO2 of the blood. So whenever there is a decrease in the pH or fall in the pH, the chemoreceptor senses this change in pH so that there is a stimulation of respiratory center. So whenever there is a stimulation of the respiratory center, the respiratory rate increases. So that due to hyperventilation, this will lead to elimination of carbon dioxide or expulsion of carbon dioxide from the lungs and lowering the carbonic acid level. So we know that there is an inverse relationship between partial pressure of carbon dioxide and the pH. So whenever there is a decrease in the partial pressure of carbon dioxide, it leads to increase in the pH. So this cannot continue for long, the increase in pH, due to decrease in pH which are sensed by the chemoreceptors, stimulation of the respiratory center, and the increase in the respiratory rate, thus decrease in the pCO2 leading to increase in the pH, cannot continue for long. The pH increased by the elimination of carbon dioxide will depress the respiratory center and rate of respiration falls. Therefore, the respiratory system responds to any change in pH immediately but cannot proceed to completion unless the underlying pathology is corrected. Whenever there is increase in the pH, this change in pH is sensed by the chemoreceptors, as the central chemoreceptors and the peripheral chemoreceptors, which causes inhibition of the respiratory center, so that there is decrease in the respiratory rate and accumulation of the carbon dioxide, so that there is increase in the partial pressure of carbon dioxide, which leads to decrease in the pH because carbon dioxide is a volatile acid. So this is how the respiratory system regulates the arterial blood pH whenever there is increase in the pH, which are sensed by the chemoreceptors, and inhibition of the respiratory center leading to decrease in the respiratory rate and increase in the PCO2 and thus decrease in the pH. But this will not prolong for a longer time unless the underlying pathology is corrected. In the previous slides, we discussed that hemoglobin acts as an important buffer in the RBC. As well, it has an important role in regulation of the pH. Just consider that the RBC has reached the site of tissue and it's carrying the oxygen in the form of oxyhemoglobin. So when the RBC reaches the site of tissue, the carbon dioxide which is obtained by the oxidation of fuels, out of which the major fraction, around 75% of it, diffuses into the RBC. So where carbon dioxide reacts with water to form carbonic acid in presence of carbonic anhydrous, and again it dissociates to form hydrogen ion, the bicarbonate. So when the hydrogen ion concentration increases, the hemoglobin gives off oxygen and diffuses into the tissues. So now we have the deoxyhemoglobin. This deoxyhemoglobin accepts the hydrogen ion and the carbon dioxide to form carbaminohemoglobin. So we have three fates for the carbon dioxide which is obtained by the oxidation of fuels. The largest fraction of carbon dioxide diffuses through the plasma into the erythrocytes, where buffering mechanisms are available to deal with it in the form of bicarbonate and hydrogen ions buffered by hemoglobin, and 15% of it reacts with the deoxyhemoglobin to form carbaminohemoglobin, and the 10% get dissolved in the plasma. So these are three fates of carbon dioxide. So at the tissue level, what happens? As the bicarbonate level increases within the RBC, to maintain the electrochemical neutrality, there is exchange of bicarbonate with the chloride ions, so that the chloride enters into the RBC. So this is known as chloride shift or the hamburger shift, which is result of carbon dioxide uptake. Thus, on the venous side, the RBCs are slightly bulged due to the higher chloride ion concentration. So, at the site of tissue, there is exchange of gases, that is oxygen and the carbon dioxide, and the hemoglobin carries the proton and the carbon dioxide in the form of carbaminohemoglobin. So, this is taking place at the site of tissue. Now, consider that the blood has reached the lungs carrying the RBC along with that carbaminohemoglobin. So at the site of lungs, what happens? The carbaminohemoglobin, here we can see the proton and the carbon dioxide along with the deoxyhemoglobin has reached the site of lungs. So here the opposite situation is found where PCO2 level is low, pH is high, and PO2 is significantly elevated in the lungs. So the oxygenation of hemoglobin takes place. So from the carbaminohemoglobin, carbon dioxide diffuses into the lungs and then expelled by the lungs. So the deoxyhemoglobin accepts the oxygen to become oxyhemoglobin and releases the proton. So now the bicarbonate reacts with hydrogen ion to form carbonic acid in presence of carbonic acid anhydrous to form water and the carbon dioxide. Carbon dioxide is expelled by the lungs. So here the main reaction which is taking place here is the carbon dioxide expelled by the lungs and oxygenation of hemoglobin and buffering of the hydrogen ions by the bicarbonate. So as the bicarbonate level decreases, to maintain the electrochemical neutrality, again there is a reversal of chloride shift in which the bicarbonate and the chloride exchange takes place, so that the bicarbonate level is maintained here to buffer the hydrogen ions. So this is known as reversal of chloride shift. The oxygenation of hemoglobin in the lungs and deoxygenation at the site of tissues under the effect of pH and PCO2 is referred as Bohr effect. The net effect is the transport of carbon dioxide produced in the tissues with the minimum change in pH and concomitant transport of oxygen from the lungs to the tissues. So far we have discussed about the first line of defensive mechanism to regulate the pH of the body fluids is by the buffer systems of the body fluids, and the second line of defensive mechanism, that is by the respiratory system, and the third one which we are going to discuss now is the renal regulation of the pH. It is an important function of the kidney to regulate the pH of the extracellular fluid by excreting either an acidic or the basic urine. The non-volatile acids which are produced in the body cannot be excreted by the lungs. So the primary mechanism for removal of these acids from the body is by renal excretion. So this is a reason why we require the renal system for the regulation of the pH. There are four major mechanisms to regulate the pH of the body fluids by the renal system. The first one is excretion of hydrogen ions, and the second one is reabsorption of the filtered bicarbonate. The third one is by the phosphate buffer system, and the fourth one is ammonia buffer system. Excretion of hydrogen ions. This process occurs in the proximal convoluted tubules. There is a generation of bicarbonate with the net excretion of hydrogen ions. The carbon dioxide generated by the cellular metabolism combines with water to form carbonic acid in presence of the enzyme carbonic anhydrase. The carbonic acid ionizes to hydrogen in the bicarbonate. The hydrogen ions are secreted into the tubular lumen. This mechanism is very effective in regulating the pH, especially when an acid load has to be eliminated. It serves to excrete hydrogen ions and generate bicarbonate, thereby increasing the alkali reserve. The reabsorption of the filtered bicarbonate is the second mechanism by which the renal system regulates the pH of the body fluids. Reabsorption of filtered bicarbonate is mainly to conserve base and associated with secretion of hydrogen ions. About 80 to 90% of the filtered bicarbonate is reabsorbed in the ascending limb of loop of Henle, and rest is reabsorbed in the distal tubule and the collecting duct, so that urine is normally bicarbonate free. The filtered bicarbonate combines with hydrogen ion in the tubular fluid to form carbonic acid in presence of the carbonic anhydrase present in the brush border of the cell wall, dissociate the carbonic acid to form carbon dioxide and water. This carbon dioxide diffuses into the cell, and carbon dioxide within the cell would again combine with water to form carbonic acid, and which dissociates to form hydrogen in the bicarbonate. So this bicarbonate is entering into the renal interstitial fluid. Here we can see that there is no net excretion of hydrogen ions or generation of new bicarbonate. There is no loss, no gain. So what are the bicarbonate which is filtered? Again, it is almost reabsorbed. So here there is no net excretion of hydrogen ions or generation of new bicarbonate, and this is a mechanism which helps to maintain the steady state of acid-base balance, but may not be effective in meeting an acid load or replenishing the alkali reserve. So there is no net loss or gain of bicarbonate, but maintains a steady state of acid-base balance. excess of hydrogen ions cannot be excreted by combining with bicarbonate. The excess hydrogen ions must therefore combine with non-bicarbonate buffers in the tubular fluid. Hence, we require the other two buffer systems in the renal tubules. So that is phosphate buffer system and the ammonia buffer system. The phosphate buffer system consists of monohydrogen phosphate and dihydrogen phosphate, which we have already discussed in the previous slides. The pK of the buffer system is about 6.8 and urine pH is slightly acidic, and thus under normal conditions the urine pH is near the pK of the phosphate buffer system. In the proximal tubule, the effectiveness of the phosphate buffer system is limited by its low concentration due to increased reabsorption of the filtered phosphate. But in the distal nephron, the phosphate concentration in the tubular fluid can be significantly increased due to the reabsorption water in excess of phosphate. Thus, phosphate represents an important tubular fluid buffer primarily in the distal nephron. The amount of available phosphate remains relatively constant even in acidotic conditions when the kidneys must excrete additional hydrogen ions. Here we can see that the filtered monohydrogen phosphate reacts with hydrogen ion to form dihydrogen phosphate. How do you obtain these hydrogen ions? It's from the carbon dioxide, which diffuses into the cell, reacts with water in presence of carbonic anhydrase to form carbonic acid, which again dissociates to form bicarbonate and the hydrogen ion. So this bicarbonate which is obtained is the new bicarbonate which enters into the renal interstitial fluid. At the same time, the hydrogen ions in exchange with sodium ions enters into the tubular lumen, which are buffered by the hydrogen monohydrogen phosphate to form dihydrogen phosphate. Hence, the urine will be acidic in nature. The excreted dihydrogen phosphate represent the major component of the so-called urinary titrable acids, defined as those weak acids in urine that can be titrated when a strong base such as sodium hydroxide is used to bring the pH of the acidic urine back up to the pH of the glomerular filtrate, that is normally pH 7.4. So, this is how the phosphate buffer system generates a new bicarbonate and secretes the hydrogen ions into the tubular lumen, which are buffered by the monohydrogen phosphate and excreted in the urine as dihydrogen phosphate. Ammonia buffer system is one of the mechanism by which the renal system regulates the arterial blood pH. The components of the ammonia buffer system are ammonium ion and ammonia. The pK of the reaction is 9.3. Ammonia present in the body fluids is the protonated ammonia ion. Ammonia buffer helps to excrete hydrogen ions and generate new bicarbonate. Predominantly, it occurs in the distal convoluted tubule. Ammonia is highly effective buffer for secreted hydrogen ions because the ammonia is synthesized in the epithelial cells of the proximal tubule and distal nephron and secreted into the tubular fluid. Approximately 60% of ammonia is obtained from glutamine. So the glutaminase acts on glutamine. The ammonia molecule is released from this, and the glutamate which is formed from glutamine again undergoes reaction in presence of L-glutamate dehydrogenase to release ammonia. So 60% of ammonia is obtained from glutamine and 40% is obtained from the other amino acids like glycine and alanine. And also, ammonia is lipid soluble and can passively diffuse across the cell membrane, but ammonium ion is highly polar and crosses the membrane poorly. Here we have the generation of new bicarbonate by ammonia buffer system. Ammonia diffuses across the cell membrane into the tubular lumen, reacts with the hydrogen ions which are secreted from the tubular epithelial cells so that it forms the ammonium ion. So the ammonia is trapped in the form of ammonium ion. So how do you obtain these hydrogen ions? This is obtained from the reaction of carbon dioxide, which diffuses into the epithelial cell, so that tubular epithelial cell, so that it reacts with water in presence of carbonic anhydrase to form carbonic acid, which again dissociates to form bicarbonate in the hydrogen ions. Bicarbonate, which is obtained, is a new bicarbonate by ammonia buffer system, so that by the ammonia buffer system we are generating a new bicarbonate as well as losing protons in the urine as ammonium ions. The first line of defensive mechanism is by the buffer systems of the body fluids, which acts immediately within fraction of a second, and then the second line of defensive mechanism by the respiratory system, which is always fast, so that within 12 to 24 hours it tries to bring the pH back to normal, and the metabolic compensation is always slow but most powerful and long-term regulator of acid-base balance. Thank you for watching this video lecture on acid-base balance. Wait for my next video lecture on acid-base disorders. Till then, goodbye. But don't forget to subscribe this video channel.